Right, so good morning, everyone. So, all right, so I want to welcome you to chemical biology and biomedical research to give you an overview of what this course is going to be about. It's going to be a bit experimental, so I hope that you appreciate that. It's not a traditional class, know that in advance. This is meant to be an immersion into research and taking information from research to develop new knowledge. And so as part of this course, we're going to be engaging in that type of principle and concept of learning so that we can produce something that will go into. So let me tell you a little bit about myself. Apparently my voice is not working. All right, so my name is Dr. Arthur Tinoco. This is the older picture of my research laboratory. I am a bioinorganic chemist in training. I do research that focuses on metals in terms of their role in biology. So looking at how biomolecules regulate these metals and using this insight for the development of metal-based therapeutics. So I'm located principally in Coconut West, but I also do research at the Western Science Research Center. You can get a little orientation about myself. As far as my availability, if there's a need to come to me for anything related to the course, I will be available on Tuesdays and Thursdays every after class in the afternoon. Although we can certainly meet outside of those hours. Typically, I'm available at campus, so we can meet whenever more convenient to you. And as you know, you already have received some email correspondence. But I also wanted to introduce you to additional people who are participating in the implementation of this course. So the first person that I want to introduce you to, one who actually brought this idea to me, there had been a previous iteration of this course that was facilitated by a professor in the biology department of several little ages. And so she wanted to bring this course back to life. And so this is Natalie Ricci. There she is. So I'm going to give you a moment for her to introduce herself to you guys. Right, Natalie. Your mic is off, Natalie.
Of course it is. Thank you all so much for taking this course. I'm really excited to bring this back to life. Like Arthur said, this is something that we have been doing for a few years now. And I'm so sorry, my little dog is making noises. He wants to come and say hi. So this is something that we've been dreaming of for a while now. And it started a few years ago. Here he is, everyone say hi. Started a few years ago when during the pandemic in Memphis here in St. Jude, we lost a postdoc. She died unexpectedly in a horrible car accident, and so we were really sad about that. And so one of the things that we decided to do in her honor was create a course and an internship that follows up the course. So I would like you all to know that we do have an internship available in the Department of Chemical Biology and Therapeutics at St. Jude Children's Research Hospital for this coming summer. I think Arthur has a QR code that you may be able to connect to at some point. There it is. So this is an in-person internship in a lab in chemical biology and therapeutics at St. Jude Children's Research Hospital for 10 to 12 weeks. We pay for all the travel and housing, and there's a $2,600 per month stipend. Applications are due soon, February 1st, and the QR code goes straight to the application site, and then there's a little short link there. So this is something that we've been doing even when the course hasn't been happening. So one of your TAs, Aixa, actually did this last summer. So if you have any questions, please feel free to ask her all about it. We've hosted six students so far, and all of them have really had a great experience. Most of them have ended up either in medical school, graduate school, or MD PhD program. So it's been a really great experience for them to figure out where they want to go with their career and help them either solidify their interest in research or decide that they want to go straight to medicine. So if you're on the fence, this is a really great opportunity for you. So that being said, I really look forward to having you all take this course this semester. And if I'm lucky, I will get to come and visit you before the semester's up, but we'll see how that plays out. So again, thank you. Thank you so much for taking this course.
All right, so next we have the program mentioned will be.
Would you like me to introduce myself or Jose?
Uh,
Or
uh,
Isa?
They keep going back and forth.
All right, so next we have Dr. Jose Reyes, who actually was an undergraduate student at our university. I had the honor of teaching him in organic chemistry. So Jose, introduce yourself.
Yeah, thanks, Arthur. Yeah, my name is Jose Reyes. I was an undergrad in the University of Puerto Rico from 2016 to 2020, right as the pandemic started. I was heading out. I did my PhD in University of Michigan, and now I am here at St. Jude's. This is my first year. I just finished my PhD, so I just started. I'm a postdoc in the Siemens Saris Lab here at CBT. So if you guys decide to join the Nancy Martinez Fellowship, you would potentially see me a lot around this area of the CBT in St. Jude. Yeah, I mostly focus on areas of chemical biology, specifically in chemically induced transcription and translation, and also understanding how synthetic gene regulators or synthetic transcription factors work. I'm really excited to be able to TA a class from my home home institution. It's pretty cool, pretty full circle moment. Yeah, I'm happy to answer any questions, and you have my email there. Feel free to ask any questions, and hopefully uh, it will be a good semester.
All right, thank you, Jose. All right, so next we have Dr. Diane Lavoie Citron, who will also be participating in this course. And I'll talk a little bit more after they introduce themselves exactly what role they're in. All right, go ahead, Diane.
Cool. Hi, everyone. I'm Diana Wicintron. I'm also a first year postdoc at St. Jude. I did my PhD at the University of California in San Francisco. My area of like research is functional genomics. If you don't know what that is kind of moving from sequence, so DNA is a string of A, C, T's, and G's, and moving into function. And I do like high throughput, and then we do this at high throughput. Genetics and severe forms of childhood epilepsy. And yeah, I'm really excited to be here. I just really want to emphasize that my path to research from undergrad to my PhD to my postdoc was shaped greatly by internships. I did summer internships in every single summer during my undergrad at other institutions and really shaped my path to be here. So really wanted to emphasize if there's any interest, even though I didn't do one at St. Jude, I would be happy to talk about that experience as well.
All right, thank you so much, Diane. The next we have, and we actually have her here in person, Aika Orta Rivera, who is a PhD candidate working in my laboratory. She's focusing on a medicinal and organic chemistry project. She will be graduating this year. A really important thing about her role in this course is that she was a Nancy Martinez Fellow. And so she did that last summer. She worked in the laboratory of Dr. C. Sorry, go ahead, Aika.
So good morning, everyone. My name is Aika Ortland, and I have been a UPR student for a long time. So I started off as an undergrad student, and I then transitioned into my PhD. And once I transitioned to the PhD, I just fell in love between the mixture between chemistry and biology because I was always conflicted because I always loved both fields. And within this opportunity under Tinoco's mentorship, I was able to work in the synthetic component, but also do some biological assays, and I expanded those techniques in the St. Jude Children's Hospital. So I really thank you for this course because I think it's a good merge of both topics as well.
Thank you, Aika. All right, so let's talk a bit about the course. All right. No, it is connection. All right, so I've handed everyone the syllabus. I've also emailed you the syllabus first. I imagine it's gonna take some time to get a little bit used to how this is going to pan out. But a few things I want to nail as far as like what is the objective or what is this course going to be about. And so, as the name implies, we're going to look at the field of chemicology. And I'm going to attempt to provide you a definition of what chemicology is. But the key thing is that we're going to use the insights from this field in order to expose you to biomedical research. And so some of you are already engaged in research, maybe not necessarily of a biomedical nature. Nonetheless, the tools from this field can be applicable beyond. So that's something that I want you to get across. So through this course, we're going to have an introduction of what is the general role of chemical biology strategies for biomedical research, provide a basic background and fundamental preparation for a career in STEM, possibly in chemical biology in a biochemical research context, but also to try to encourage some of you to pursue graduate studies, whether it's in chemistry, biochemistry, biology, just to like take some of these tools and be able to translate them into your later career objectives, pursuits. So provide a basic level understanding and familiarity of laboratory safety, equipment, techniques, procedures, while using cancer-related case studies and publications to teach how to read science, how to write scientific manuscript, and also to present chemical biology research. And so that's going to be a really critical part of this course is the engagement with reading. I know that some of us don't love to read things, but we're going to try to learn how to more critically dissect scientific literature, particularly primary literature. And so through what we're going to implement in this course, we hope to get that so that it's something that's more enjoyable. Okay, so what are our key objectives? So what we want to do is to introduce and apply some of these research practices in the chemical biology field so that we can look at a targeting of cancer. So our main course project is going to be related to cancer. We're going to talk a bit about that, but we will have a specific lecture that will define exactly what this project will be. So students will apply chemical biology concepts to real-world questions and problems related to biomedical. The course will give students the ability to learn reading and writing in scientific context, and you will all participate in the writing of a review article centered on the application of chemical biology tools for a cancer drug. And again, we will look more into that as we progress through the course. So what exactly is the format? And so we're going to see that we have some traditional lectures. This is a course that's taught in conjunction with experts from St. Jude's. And so these experts coming from the research hospital, in addition to the Graduate School of Biomedical Sciences. So it will not just be myself who will be facilitating the course. You're going to have experts from there. In addition, you're going to have Jose, Diane, and Isaac who are going to help facilitate aspects of the course that have to do with the discussion and the section of the literature. So this course will have, I said, the combination of traditional lectures, and that's going to be at the minimal part of the course. The majority of the course, we focus on more of the application or listening to outside experts talking about the different areas of the field. So the PowerPoint slide will provide visual aid to emphasize fundamental concepts and their research application. Every week will consist of one formal presentation lecture and one journal club discourse. So the journal club, we'll go into that in a moment. The lectures will be offered by several faculty experts based on their research expertise. The journal club will be facilitated by the course CAs and myself to some extent. And we'll follow a student interactive pedagogy. All right, so this is try to break this down. What exactly are we talking about? So when we're talking about a journal club, we're talking about we're going to have an article or two that's going to be signed, or possibly selected by one of you guys. And so what we would do is we will talk about the manuscript, and we're going to look at it from a few perspectives. One is to look at the tools that are actually being used, because sometimes we can gloss over the experimental components just to highlight what was accomplished. But the thing is that we want to understand what are the actual tools, how they work, and what kind of insight can you actually get. That's important. What insight can you actually get rather than what they're telling you to produce? Because then you know what are the limits that can be achieved with a particular tool, and then you can dissect figures much more, I would say, in a much more constructive way to see do you actually concur with what's actually being described in those figures. All right, so that's the component of the journal club, and we'll have that on a weekly basis. As far as the material for the course, I said one main textbook, but actually I provided you with two textbooks just because we're gonna take excerpts. We're not gonna actually use all the material. As the majority of the course is going to be based on material presented by outside researchers. And so we will be using a lot of the material that's based on the work that they're doing. So the primary literature. That'll be the majority of the literature that you have in this course. But these additional sources will help us to fill in some of the blanks in order to have a better understanding of certain things with regards to concepts or techniques. I prepared a website where we will be able to upload the material from the course. I already uploaded this lecture, so you can go and download it later on and review the material in this lecture itself. Now, as far as subsequent lectures, depending on who's facilitating them, they may or may not be available ahead of time. But as much as we can, we'll try to have this material available to you as quickly as possible on the actual website. Okay, take a pause. Questions so far that we have? You good so far? All right. So as far as the actual breakdown of the course, there are no exams. The whole idea is to try to get you immersed into understanding at a basic level chemical biology and tools that you can use for different applications. And so the way that we're going to do this course is that the assignments, and that has to do with the journal clubs, will count for the 50% of the course. And your participation, that means you're engaging, that's the student interactive pedagogy, which is that you're going to play a big role in terms of looking at the literature and seeing what are we actually learning. That will be the actual course project, which will be a manuscript, but also coming to my presentation, where you will give a presentation of the research that you did at the time. So because of that, this course is essentially a pass-fail course. So either an A or an B. So as long as you collectively earn a score that's greater than 70%, you earn, right? And if you earn less than that, then this will be the grade that's assigned to you. So I don't expect anyone generally have any knowledge of who you are. You get really scores, and so I expect that would be a relevant grade. Okay. All right, so let's get into it. So what is chemical biology? And so I was trying to get a definition, and I was looking at the literature, but this came out in 2015, where they asked a lot of the top researchers their definition of chemical biology, because the premise of this particular, it was, I would say this is a commentary or opinion piece, was that there didn't seem to be a one defining definition, that's the right way of saying it, one definite definition. And so they were trying to get a sense of what people were viewing chemical biology as. And so if you read some of these quotes, it's actually kind of funny, but some of them even say like, when you know, let me know. So I want to give you, this was in 2015. So Carolyn Vitosi, Stanford University. Definition: chemical biology is an area of research in which chemical and biological concepts and tools interact synergistically in the pursuit of new discoveries or technology. And then Christopher Chang, who was originally was at Berkeley, has recently moved to Princeton University. Chemical biology involves viewing the world around us, the living organisms and their environment, the lens of a chemist. And taking advantage of the unique ability of chemists to not only study, but also create new forms of matter at the molecular level for a societal effect. Okay, so we have one that's more of, I would say, a formal definition, one that's a little bit broader in terms of perspective. And so let's try to get a sense, because I would say it's still not clear. And I would say probably at the end of this lecture, it may not be still clear, but the whole point of this course is to develop our own definition for chemical biology. All right, so a working definition. Using chemistry to advance a molecular understanding of biology at the atomic level, right? So this is what distinguishes this field from biochemistry. Sometimes biochemistry tends to look at molecules. Sometimes you actually do biochemistry. You probably talked about hemoglobin, and you talked about the different units, and maybe a little bit about the iron, but not so much. And so sort of the atomic level gets lost there. You may talk a little about amino acids and what role they're playing as far as maybe facilitating, control of the pH, things like that. But this field tries to really look closely at the atomic level and the bonds that are there, those interactions that help to, facilitate life, but also what we can learn from them in order for us to develop techniques that we can use to probe these type of interactions to understand how things function, and then to use them for different applications. It also is using biology to advance chemistry. So essentially, you're applying techniques from one field to address problems in the other field with potential real-life applications. And that's going to be the working definition that we're going to use, and then as we develop the course and we hear from outside experts, we can better understand what this field really is. All right. So let's get a bit of a historical perspective. I will say this is not necessarily the beginning of chemical biology, but certainly what I'm going to talk about right now is a very influential moment in terms of history that led to development of different fields, chemistry, organic chemistry, chemical biology. And that is the synthesis of urea. It's a pretty simple molecule. This was a process that was done by Wörtler in 1828. Now, why is this an important experiment? Well, what do we know about urea? Does anyone know anything about that particular molecule, urea?
It's a waste molecule.
As what?
It's like what they say, part of waste.
Part of waste, but where is it coming from?
It's found in urine.
Found in urine, okay? So it's coming from a living organism, right? Right, very important. So it's a molecule, the biomolecule. And so why this is an important experiment is because it's an experiment in which a biomolecule was synthesized. And by today we were like, oh, well, that's fine. Again, it's a big deal. But back then, it was a very big deal because what was trying to be done at that time was this experiment, this chemical reaction here, where you have a silver salt interacting with ammonium chloride, and the goal was just basically to do an exchange of those metals, okay, by the application of heat, or some sort of energy here. Well, what ended up happening was the following, where this molecule was a biomolecule, and that was something that really shocked the field because this is a biological metabolite that's found in Europe. The thing was, at the time, the concept of vitalism was at play. What exactly is that concept? So this is a 19th century theory that these type of molecules, these organic molecules, some living things needed a vital force in order to be made. And so you couldn't do this necessarily outside of that force. However, what this experiment showed was that you can actually use chemicals and energy in order to produce a biological product. And so it demonstrated that a biological molecule could be produced by pure chemical. And that's what was so radical, that it actually broke down this concept of vitalism, the idea of a vital force in order to be able to produce this type of molecule. And so here I'm going to show you a collection of different key types of examples of chemical biology and how they really have shaped the field. And so again, this is not meant necessarily as an order. So I skipped a number of things that are important historically, but just want to highlight a few things. For instance, Joseph Priestley. So this is dating into the time of 1733 to 8th century. This is incorrect. I'll fix it. Experimented with gases like nitrous oxide, and the goal was to see how do living organisms, in this case mice, react to the exposure to these gases. So it was to see the biological effect. We're using chemical things and trying to see how these systems react to them. Then we have Ehrlich, who in the 1854-1915 developed the concept of magic bullets. So what exactly was he referring to? So the idea of using a molecule to try to treat something, so use the molecule as a therapeutic. And so from the discovery of this particular salvarsan to treat syphilis, that was actually a really big discovery, because now we're starting to invent molecules, develop molecules that can actually be used therapeutically. So that essentially helped to pioneer chemotherapy to target specific pathogens. And you also have enzyme inhibitions as a probe of biology. So an example of that is the use of penicillin. And so what is penicillin? I think you guys have probably been exposed to penicillin. Some may be actually allergic.
I mean, that's an antibiotic.
It's an antibiotic.
As a beta.
Yeah, so as an antibiotic, but also can be used as a chemical inhibitor of bacterial cell wall biosynthesis. So the way that you can try to use a molecule to interrupt something to try to understand that mechanism. There's also affinity labels and active site probing. As an example, the use of diisopropyl fluorophosphate in DSP to label serine residues and serine proteases. So these are the serine amino acids in those particular enzymes. And so the idea is that you're going to use a probe potentially to try to understand how that particular enzyme is working. Chemical control of gene expression. So in this case, the use of small molecules like lactose, IPTG, to regulate gene expression in E. coli. So this is an example of a small molecule that's being employed to control genetic circuits. So these are really important fundamental discoveries that we've been able to take advantage of and help to really develop the field of chemical biology. Okay, so what we're going to do is we're going to talk a bit about, life itself and to try to understand what is chemical biology, what are the driving forces. So one of the principal things is the central dogma of biology. This was from Francis Crick. So I'm going to read what this is, and then we're going to try to decipher it. So the central dogma states that once information has passed into proteins, it cannot get out again. In more detail, the transfer of information from nucleic acid to nucleic acid or from nucleic acid to protein may be possible, but transfer from protein to protein or from protein to nucleic acid is impossible. Information means here the precise determination of sequence, either of bases in the nucleic acid or amino acid residues in the protein. Okay, so here we're talking about a transfer of information, and we will talk a bit more in the subsequent lectures a bit about these particular molecules so we have a better understanding of what exactly is being discussed here. But I want to give an overview of this dogma. So from this statement, we have what we call a deterministic viewpoint. And this deterministic viewpoint is looking at how everything is synthesized in itself. So it's highly regulated. That's essentially what that implies. So here is essentially the breakdown of this dogma, where we're looking at DNA and how that's basically what's going to dictate what comes as a consequence of what's built into the sequence of DNA. So we have that you can have DNA replication through the use of an enzyme, DNA polymerase. So DNA can then produce DNA. You can then go from DNA to produce an RNA. And this is with the process of transcription, which we'll discuss more later in the course. And so essentially going from a DNA molecule to an RNA, where the sequence of information is coming from the DNA itself. And so that's the idea of how you transmit information. There is the possibility of going from the RNA to the DNA using a reverse transcription process. But essentially still, you're getting this information initially from an actual DNA molecule. All right, so now we're in RNA, and then we have the process of translation. And this process of translation occurs through the use of ribosomes. So essentially now you're translating the information that's within the RNA in order to produce proteins. Okay? And so here it's as built in according to the dogma, seen as the endpoint. But in reality, that is not the endpoint. We'll talk more about that later on. But the premise here is information starts from here and it goes to here, and it can go backwards. That's what the idea is with regards to the central dogma. One quick thing is that with RNA, you can replicate RNA, but that's actually pretty rare in terms of seeing that in terms of organisms. You can also have RNA-dependent RNA polymerase in order to have a different strand of the sense RNA. But again, it's a transfer of sequential information is viewed in a very deterministic fashion. So when we're looking at the DNA, we're focusing on the genes that are going to be transcribed. That essentially is defining what we call it in the world of omics, right? The genome. When we're focusing on RNA in terms of being able to transcribe DNA into RNA, essentially what we're focusing on then is that collection of the RNA, which would be your transcriptome. And then the production of the protein through translation will be essentially the collection of proteins that we're going to refer to as the protein. And so that's actually really important when we're looking at systems biology and the omics world, how we can learn information from them, what specific molecules we're going to be focusing on. Okay, so now let's start to dissect the idea of the central dogma. And one of the things that we can focus on is the reality that when you produce proteins, it actually is not the endpoint for many proteins. Okay, so why is that? Why is that not the endpoint? Well, it turns out you can have what's referred to as a post-translational modification or post-translational process. All right, so what does that actually mean? So here I'm going to give you an example of what that means, where these PTMs, they can happen via, let's say, like a chemical process where your protein might have an amino acid become oxidized. Often, what you see is an enzyme plays a role in terms of modifying this protein. So an enzymatic covalent modification of the protein itself. So where can this actually happen? So it can occur on the amino acid side chain of the protein. It can occur at the two ends of the protein itself, right? When we talk more about protein and structure, we'll get a better understanding of what that specifically refers to. So primary post-translation modification, what they achieve is to functionalize the protein. And oftentimes, this change is important because it enables the secretion and also the solubility of these proteins. That's a critical property, especially for certain proteins that may, let's say, serve a transport process. Unless they are positioned in the membrane of cells and their role is literally to stay there and facilitate transfer in that modality. Okay, but what about additional modification? These subsequent PTMs, what are they achieving? So they can lead to further specialization of function. They can lead to the generation of bioactive peptides, or they can just be part of the natural process of degrading a protein into this constituent components. So here's just this cartoon representation where we have this sequence here, I just drew it as blue dots to represent these amino acids that are in the sequence define that protein. You have your primary post-translation modification that gives you what is the mature protein. That's the protein that is created. And then you have proteases, which as the name implies, those are protein enzymes. And the function of this is to then cleave these proteins into smaller proteins or into sequences that are essentially like a protein can be seen as just a very big peptide. All right, so you can have bioactive peptides. Okay, so they have their own functionality that are distinct from the parent protein, or you can just have what is referred to as a degradation, proteolytic degradation. That is basically breaking down the protein into, say, peptides or amino acids, and they serve as a source of nutrition for the body. And so again, if you add the bioactive peptides, well, then you can have further processing of their protease or peptidase. So those are enzymes that are specific to peptides that will break them down. And then again, this is part of the proteolytic degradation. Okay, so it turns out that synthesis by cells is not purely determined. So as I was mentioning to you, this idea of DNA dictates RNA dictates protein. Well, those processes that are, for instance, a post-translation modification shows that you're going to get a diversity of things that can form and that can influence what is actually synthesized within cells. So while DNA may dictate the transfer of information in cell synthesized molecules, these molecules that are produced participate in processes that are not dictated by DNA. And that can result in the production of other molecules. So here's a map of chromosome strands. And so what we have is the identification of different genes. We're not going to really get into the details of this just yet. Essentially, the arrows are basically dictating the order in which the transcription will occur. But what these different arrows are showing are genes that can be transcribed, that those transcripts can be translated and produced. Okay. So you can get a number of proteins that can ultimately be produced from what's represented here. Now, the thing is, once those proteins are produced and those proteins have different functions, now at that point, this chromosome, the DNA, has no control over it. Those proteins will operate independent of what was originally placed into them to be just controlled. So that's going to be critical to understand because you're going to have a number of processes that are going to facilitate the diversity. So you have transcript copies that are determined by transcription factors, different resultant mRNA derived by RNA splicing, PTMs that can alter protein structure and function, and as a consequence of altering the structure, obviously potentially the function, can lead to different types of metabolites that are being generated. So that central dogma that I showed you, I didn't even include that the proteins can produce metabolites. Something that we'll talk a bit more about later. All right. So it turns out that evolution, this concept, is actually a founding principle of chemical biology. So a key objective of chemical biology is to organize and try to simplify knowledge about the living world in order to use this information to guide experimental design and address mechanistic problems. So we can break it down into two major steps. There's a lot more to it. Let's try to simplify that in line with the principle of chemical biology. So create a diversity of specialized organisms and molecules. So what does that really mean? Well, you can try to evolve organisms, so there are ways to do so. Okay. For instance, there are a number of people who try to, knock out genes from mice, and you get a different variant of a mouse. So it becomes an iterative process. So what does that mean? You might have to do a few cycles in order to be able to achieve the end goal you're looking for. So that's in the evolving organisms, or you can have a combinatorial shuffling of molecules in order to generate a desired biopolymer, protein, the acid you're interested in, that can be useful for something. So you create this diversity of organisms and molecules. Okay, what's the next part of it? Well, then sifting through what you have created to find the fittest. Okay, fittest for what? What exactly does that mean? Well, that means that you have a certain thing that you're trying to generate these things for. And so you're trying to determine what of what you've created is optimal for the application you're seeking. So you sift through using a high throughput activity assay or some other method to identify optimal molecules for a particular application and function. Okay? So this is basically how we can look at what is a working principle for chemical biology in terms of how we can utilize the information that we either obtain or exist in order to create things that can be used for further programming. Right. So with that in mind, I want to direct you to what has been where the field is really directed to in terms of a real life application. That is in the area of drug development. And so here we're going to look at this idea of how drug development has evolved and where chemical biology has really made its actual mark. So I'm going to give you an overview of just the process of how a drug in the United States is compounds produced, it is actually assessed, and how it can actually reach the clinical market. All right, so in the United States, the regulatory federal agency is the Food and Drug Administration, and there are some agencies like that in other countries, but not all. So the FDA Center for Drug Evaluation and Research is the office responsible for conducting the evaluation of all safety and efficacy studies performed on drug candidates. And so here I provide you a map that kind of goes through this process of the clinical stage, how something that advances through here and shows promise as a drug, what you need to do in terms of applying for what is referred to as a new drug application. And if something advances to then be actually able to enter into the clinical space, what you need to do in order to promote or to try to help implement the use of that particular drug. All right, so let's go through this. There's some of this is probably going to be things that you're very familiar with, but it's important so you can see where chemical biology is really making its way. So it turns out that a small percentage of new compounds actually survive all the regulatory hurdles that are involved in terms of developing a drug. And so what we have is first, what is the preclinical phase of drug approval processes, and that's up to 2 years in terms of how much time it can take to really assess the potential of a compound. So this phase requires the use of an appropriate in vitro model. But for instance, if we're saying we're doing something that's developing a drug for an anti-cancer purpose, you want to look at drug candidates and test them against a variety of, say, human cancer cell lines and see what type of behavior they have. That could be your method of cutting off, filtering compounds which is really not showing much problem. All right. Then you have the most promising compounds are tested against appropriate animal models. So if we're talking about cancer, with respect to an anti-cancer agent, a suitable animal model might be a mouse that has been implanted with a human tumor, known as a xenograft, and then you try to assess whether the compounds that have shown promise actually have beneficial activity and also low toxicity. If the compounds exhibit desired properties in the animal model, then their pharmacology, their formulation, so how you prepare them, solubilize them, and their toxicology are examined. So these studies are going to reveal some critical things. We're going to reveal the best formulation for administration, will reveal most effective dose to apply, the frequency and method of administration, oral or something intravenously, how the agent is actually metabolized, because those metabolites are important as they are going to be our understanding of how there's one, if at all. Is there any actual toxicity that's produced because of these actions? All right, so let's say that we make it through all this and we find that a compound is promising. What do we do next? Well, the next thing would be the application of an investigation of a new drug. The sponsor of the drug candidate, which can be a pharmaceutical company, academic institution, research institution, so forth, will have to file this with the FDA. And so what information needs to be included in this? The chemical structure of the drug, explanation of how the drug may operate in the body, details of toxic effects on animals, and details as to where and how clinical trials will be conducted. Okay. So the clinical trial, now we're moving to the area where we're now going beyond those models, right? And we're now going to apply them to humans. And so it's very important that all this information is documented in such a way that it's that we know how we're going to actually implement this into. Okay. So the sponsor must provide a plan for clinical trials which describes the following. What are the institutions that are going to be involved in this trial? What are the number of people that will take part of it? And what are the exams that are going to be conducted? Then you will have what is the institutional review boards of each of the sites of the trial. And so the board consists of health professionals, they can consist of clergy and consumers. So not just experts that are informing. These are people that are interested in terms of human health. And so that's where you've got people from different areas that are formed these particular groups. They must evaluate the clinical trial plan, and they must ensure that people involved in the study will not be exposed to undue risk. That's why it's critical to have a lot of information beforehand, before applying them to people. All right, so we move it from the preclinical phase, we're going into the clinical phase, and so now we have different phases of trials. So let's talk a bit about them. What do they consist of? So you have what is referred to as the phase 1 clinical trial, which can take up to 7 years. So what does this particular phase involve? It involves a focus on safety. So what does that mean? So we're not necessarily looking at efficacy. We're looking at, can the human body tolerate it? Okay. And it turns out that, even though I said 7 years, is actually the shorter in terms of this assessment. It involves a small number of mostly healthy volunteers receiving the compound. The goal is to try to understand the side effects of the drug and reveal how the drug is metabolized and excreted. So all this information in terms of how our body can actually handle this actual compound. Then we move into the phase 2. So here now we're going from safety, assessing that, to an assessment of how effective the efficacy of it. So now we're going to ramp up the number of people involved. We're involving hundreds of patients with a particular condition, an ailment that we're trying to target. Compare response of patients to the drug with similar patients receiving alternative treatment or placebo. Monitor safety and short-term side effects. But again, we're going to look at efficacy, but always we're going to look at safety. That's going to be a critical thing to always assess along the way. Then depending on the results leads to discussion with the FDA regarding, do we go further or just the endpoint? And it turns out that many compounds either fail at phase 1 or they may fail at phase 2, where perhaps they don't show too much toxicity, but they're just not effective enough. And that's going to be another filtering point. Okay, now phase 3. This is the longest and most expensive phase of the clinical trial. Now we're going to have a lot more people involved. So this can involve up to thousands of people in clinics and health centers. The participants consist of predominantly patients, but also include some healthy volunteers. The participants receive the drug candidate or a standard approved drug, so there can be a comparison with what's a potential, let's say, gold standard in the field or a placebo. And already you're going to focus on safety and efficacy. And safety is always going to be underlying all this. You might involve what is referred to as the combination drug therapies. I mean, you might be mixing more than one drug, and maybe also trying to assess the order of administration, maybe co-dosing or treating one at a time to see the effect. It actually features very high failure rates, so about 50% of what actually tested at this level, due to a number of issues, poor pharmacokinetics, lack of absorption, the body just doesn't retain much of it, rapid metabolism and excretion, drug-induced death. You see, that's none of this is designed. All right, so a compound makes it through here. Now we are ready to see, to move it towards getting closer to clinical applications. But here we applied for the NDA. And so what does that involve? So that's been found after successful drug trial completion, reports all the information that was obtained in those trials, the efficacy, the safety, and the key thing is that you need to demonstrate the benefits of using outweigh the risk. Describe the manufacturing process and why is that important? Because you want to have something that's relatively easy to be made, not super expensive, and you also want to have relative ease of synthesis, high purity, and high integrity. So all that, where chemistry is very important, that to be able to show that this can be easily made. The FDA evaluation takes up to a year. And so here is where they produce what are called the drug labels. And so we see labels on pick up the bottles from the pharmacy, and also there's a facility inspection. And let's say that it makes it to that point. Okay, so now you're in the post-marketing in phase 4. So the total time from discovery, preclinical to final approval can be 8 to 10 years, but actually if you add in all the preclinical consumer prior to that. And so additional factors must be considered in the movement of a drug to the market. Those post-clinical trials, what does that actually mean? So the manufacturer of the drug must submit periodic safety updates on the drug to the FDA because not all potential problems are revealed during the clinical trial. All right, so how does that happen? So I'm not sure if you've ever had a drug that you just entered into the market. They often will give you that drug and then they would assess your blood at a certain amount of time. And that information is information that gets fed into here. Because that's additional trials that are being done to see, in people in longer term use, how is that drug affecting the body, in addition to whether it's actually having the effect that's information for the updates provided by physicians who report adverse side effects associated with the drug directly to the FDA using a standard protocol. So that's essentially what I was referring to. The identification of new risks can lead to revision of treatment protocols, and in a few cases, remove of the drug from the market. And that has happened in a number of cases. All right, so let's look at this from the perspective of holistically, all that's involved. So in the US, you can patent a compound, and so often a drive for this is to patent and to make money off of the drug that you're producing. So a patent can last 20 to 24 years. 1/3 to 1/2 of this time is consumed in the testing of the compound. Right, so a good portion of the lifespan patent is just the assessment. So the R&D cost for a single drug can be about 1.3 billion in the US and 1 billion in capitalized costs. The reason why drug prices are so high is because companies want to be able to make a profit. So you want to be able to make enough money that goes beyond the cost that was invested into the assessment of the company. Unpatented drugs, and there are people that do assess unpatented drugs for potential use, people, but there's a lesser financial incentive, okay, and are funded by public or philanthropic funds. So what does philanthropic mean? Well, that means people that give a donation to support the trying to study something for an application. All right. So it turns out that chemical biology is leading the way for new drug development. And how is that? Well, in the United States, there's been a lot of changes in terms of the development of drugs. As you can see, there's a lot involved in it. You want to try to streamline this. And there are ways in which you can make it much more efficient. So here, what has happened has been an emergence of translational physiology and personalized medicine, or also referred to as precision medicine. So translational physiology is the examination of biological functions from molecules to cells to organs to population. Just to understand how the body is being impacted to try to get a better assessment of the potential of the compound. So this change was stimulated by the introduction of key biomarkers. We want to identify certain things that are a predictor of an ailment that can indicate to you if things are getting better or they're getting worse. So these are using as our reference. And so sponsored by chemical biology platform, and that makes it distinct from older approaches, where older approaches were trial and error. You made a lot of compounds, you just test them to see what effect they have, and then you hope that something comes out. Okay, some of that you may see that maybe in the academic space, but more and more in the space that's trying to get things that human use, you have more of this type of platform, which is a focus on targeted drugs using, information from the omics field, mechanism-based approach to advanced clinical medicine. So less trial and error and more targets. We have, protein that we want to target, so we make them that will interact. So here's just a visual, and I took it from this review article, which reviews the growth of chemical biology in the actual drug development space. So basically what we're seeing here is how there's an assessment of the physiology, the body, how it's getting impacted, and where 5 different components are really feeding into. So you have the components to do with the biological context. You have the target identification and validation. So if you're trying to target something specifically, you look at the links from molecular to system biology. You also have a role of tools that are getting developed in order to assess how the body is getting impacted. So let's say that you have identify a biomarker, well, you need to have some sort of way to be able to assess that biomarker in order to measure it, in order to see what's happening to it. And then that feeds into drug discovery and therapy. You have these different contributions for this idea of a translational physiology. So this is the 6 key elements of the chemical biology platform. One, which is the identification of chemical parameters, the biomarkers, that are signifying a disease state. That's going to be our main criteria that we're going to be focusing on. Then you have a selection of measurement systems that will reflect cell and tissue totality, so some way of assessing how the body is responding, but also tracking the biomarkers. Animal assays to determine in vivo efficacy and safety. And so that still is in place, that's not getting eliminated. Human disease models that have the biomarkers that can be measured. Demonstration of desired change across all selected models. So that's obviously going to be critical. And linking changes to clinical benefit. Okay, so how is the body responding? And is it actual potential for a clinical application? And so what are the advantages of this platform? Where we're foregoing this trial and error, and we're having this more structured approach. Well, there's a more of an effective interdisciplinary collaboration. So this is where we have your biology and your chemists coming together and really helping to, you have the insight of how this body functions, you have the insight of those people that know how to develop tools coming together. You have enhanced drug screening capabilities, improvability to understand complex biological structures. As a result, better clinical trials in terms of the design execution. And why is that? Well, if you're trying to look at key biomarkers, you develop a compound that targets those, well, then you can select those populations that have that would potentially benefit from that assessment. So it's not just generally testing someone with an ailment, we're looking at an ailment that includes those biomarkers. And then enhance economic implications and the consequence of this is much more streamlined. There's more to it. There's other things that are contributing to what's helping drug development become much more streamlined. They can have, situations where you have an emergency case where you need to, due to an emergency situation, approve something, at least for use for a temporary time. But chemical biology is really playing a big role. So I'm going to leave it here in terms of how chemical biology is really leading the way in actual real life use, where it's playing a role in drug development. As the course will be directed towards looking at the use of chemical biology tools, but also how we can target cancer. So here I want to show you, this is something that was taken from an article in 2024, where people are looking at now what is chemical biology from their perspective. Again, I don't intend that we have our absolute definition of chemical biology, but let's look at what this person said. So this is Albert Entleman. Chemical biology is a scientific discipline born from multidisciplinarity. Okay, so here that goes beyond just chemistry and biology. This multidisciplinarity is precisely the biggest strength of the field. And what fascinates me the most about chemical biology, conveying the message that no individual discipline is better, and that the biggest discoveries only occur when we dissolve the hard boundaries between chemists and biology. And then the last one. This is from Nataran Jakinam. Whenever I think about chemology, I recall the quote from Nobel Prize winner in chemistry, Ralph Kaufman. If there were no molecules, there would be no toucan feathers, no perfume of lily of the valley, no tricolor, no paper, no ink for the score of a Dostoevsky opera, no life at all. All that makes life beautiful has a molecular basis. Not only do molecules make life beautiful, but they serve as powerful tools to understand and predict biological processes. Chemical biology is an interdisciplinary field that combines ideas of chemistry and biology to advance medicine physiology, and rare diagnostics of life. All right, everyone, I'll see you next.
También le consiguen un hermano como para yo cambiando de y no solo me lo terminé dando cuando estaba yo pienso buscar un cafecito.
Go to the attack. For our host.
Sí.